Muscles control all movement in your body, whether it's intentional or not, so they're a very basic part of your everyday life. But when you look closely at them, you'll see that they are way more complicated than you would expect. We know that muscles do voluntary things like walking, writing, and wrestling, where you're conscious of the action and you're making the decision to do it, but they also do involuntary actions like digestion, your heartbeat, and your reflexes, where you don't have to think about it for it to happen.
You can't stop involuntary muscle contractions no matter how hard you try. To showcase this, we did an experiment to see if we could keep ourselves from blinking. When a styrofoam ball is tossed towards our faces, the results showed that through all trials, there was no way we could prevent the contractions in our eyes from happening.
There are 3 types of muscles: skeletal, cardiac, and smooth. Skeletal muscles are connected to your bones, and most skeletal muscle contractions are voluntary. They are striated, or have stripes, and they have sarcomeres, which we'll explain more about later in this video. On top of doing all of your voluntary movements, they also help with your posture and position, and help stabilize you.
Cardiac muscle is also striated and has sarcomeres, but it's involuntary. Cardiac muscle is found only in the heart, so it's the muscle type responsible for your heartbeat and blood flow.
Similar to cardiac muscle, smooth muscle is completely involuntary, but unlike the others, it's non-striated and has no sarcomeres, which is why it's considered smooth. It's found in hollow organs and helps you do things like digest food.
Skeletal muscle tissue and cardiac muscle tissue are both striped under a microscope, but with skeletal muscle, it just looks like long cylinders, whereas with cardiac muscle, the cylinders appear to branch off towards each other. There are also intercalculated discs in only cardiac muscle.
Under a microscope, smooth muscle tissue just looks like cells mushed together. There are no stripes or striations or gaps or anything. It's just a blob of cells. So that's how you can identify smooth muscle tissue. In class, we had the opportunity to dissect a rat and look at muscle in different areas of the body. And so one place we took muscle from was the intestine, and it looked like this. It was very mushy, wet, gross, smooth muscle. And then this is the heart. If you ignore the blinding light in the middle, you can see the lines of cardiac muscle. And then we also got skeletal muscle from the thigh, and it was also with striations, and you can see the muscle fibers. Skeletal muscles are made of columns inside of columns inside of columns, so it's tough to understand at first glance. So we tried to make a model to help make muscles make sense. Skeletal muscles are connected to your bone by a tendon, which transitions into the epimysium or saran wrap, which is protected by deep fascia or paper towels. Below the epimysium is the paramysium or tortillas, which surrounds fascicle, which we used celery to represent. Fascicles are bundles of muscle fibers.
Each fascicle is made of lots of tiny strings. Each individual string is a muscle fiber.
If you zoom in even more on the individual muscle fiber, you'll see lots of smaller columns. These are myofibrils, and they're surrounded by the sarcolemma, which has blood capillaries and motor neurons.
Your muscles contract by reacting to electrical impulses from nerve cells. In a nerve cell, sodium and potassium ions are constantly bouncing in and out of the axon, which maintains the electrical potential that allows nerve impulses or action potential to travel through the cell.
When action potential reaches the axon terminals at the neuromuscular junction, it triggers a release of acetylcholine across the synaptic cleft and then into the receptors on the sarcolemma. This adds positive charge into the muscle fibers and generates action potential that travels along the sarcolemma and down the T-tubules, and the sarcoplasmic reticulum releases calcium.
In myofibrils, there is a thin filament and a thick filament. The thin filament has actin with the binding sites, which are hidden by tropomyosin when relaxed, and troponin. The thick filament is made of myosin and has globular hits. The calcium from sarcoplasmic reticulum binds to the troponin on the actin, which signals the tropomyosin to move out of the binding sites on the actin. When binding sites are revealed, myosin, energized by ADP plus P, binds to them, forming a cross bridge.
After this, the myosin head then moves toward the middle of the sarcomere, sliding the actin with it, and releases ADP plus P. This is the power stroke. Once ATP binds to the myosin head, it detaches from the actin and returns to the original position.
An experiment was done where a muscle fiber was put in different solutions with either plain water, salt, ATP, or ATP and salt. The muscle fiber only shortened in the solution with both salt and ATP, which shows that muscle requires both of them to contract.